Bismillahirrahmanirrahim and Assalamualaikum, Salam Sejahtera. This is my second presentation on subject STBP 6 through 6362 pengisyaratan dan pengawal aturan perkembangan sel. After correction from draft 2, I change the title of my review paper, which is Overview of Abscisic Acid ABA Signal and Pathway on Plant During Drought and Heat Stress. Before this, my title is a general overview of abscisic acid signal and pathway on wheat. So after check by Prof. Zamri, Prof. Zamri suggests to change the title just overview on plant only. So this is the content of my presentation and review paper. I start with the abstract and then introduction. The third one, impact of drought on plant growth yield. And then the main content is the phenotypic and physiological changes to abiotic stresses in plant, which are osmotic stress and temperature stress. And then overview of abiotic signaling pathway, which has involved osmotic stress perception and transduction, heat perception and signal transduction, and then stress signal transduction pathway, a common signal and pathway. Lastly, abscisic acid, a central regulator of abiotic stress tolerance in plant. Lastly, conclusion and future prospect. Okay, why I choose this topic? It's actually related to my master thesis. My master thesis title is Kesan Suhu Melampau dan Tegasan Air ke atas peringkat perkembangan bunga dengan menggunakan padi sebagai modal tanaman. Since I cannot find a more review on original paper regarding signaling and pathway in rice or paddy, so this review paper will be a general overview on plant. But I'm focused in abscisic acids, which is ABA is one of the most important hormone in plant response in abiotic stress, especially heat and dry. For the abstract part, I summarize of general role of ABA in plant, the role of ABA in plant, and then a response of ABA in transduction signal at cellular level. For me, it is very important to understand the mechanism by which plant respond to environmental signals and how these signals are transmitted at the cellular level. And then I mentioned about some functional components related to ABA, hormone, and other signal. And it is important also to understand the signal transduction to the stress experienced by the plant, as this process is very important for the survival of the plant. So in this discussion and review paper, I will consider the general features of the abscisic acid hormone transduction signal in plant against the drought stress. For the introduction part, I explain a little bit about a general overview on dry conditions. Dry conditions such as drought affect all physiological processes of the plant. By 2050, it is estimated that over 50% of the world agricultural lands will face drought. Among all the abiotic factors, drought is one of the greatest threats to the agricultural industry. So drought and heat are the abiotic stresses that greatly affect grain production. So effect of abiotic stress, especially heat and drought stress in agriculture production, I mentioned at early stage of introduction part. And then I mentioned about the effect of heat and drought on ABA response in plant, physiologically and morphologically. And then a drought of heat stress is the worst abiotic stress among all abiotic stress in crops, as I mentioned before. Drought and heat abiotic stress that greatly affect grain production. And then lastly, I mentioned about the stress, abiotic stress that disrupt the productivity of the crop either individually or in a combination form. So generally, on introduction part, I mentioned about the impact of drought on plant growth and yield. This is some literature reviews or an argument in my review paper. Phenotypic and physiological changes to abiotic stress in plant. So there are two types of stress in plant during abiotic stress. The first one is osmotic stress. What is osmotic stress? Osmotic stress is the degree of intensity and duration of osmotic stress in the plant determine the severity of cellular damage, and it will affect the growth of the plant. Any changes in osmotic phase of cell such as temperature stress can cause changes in plasma membrane. High temperature influence plant metabolic process such as photosynthesis. The degree of male fertility pollen is also influenced by the level of ABA in the anther. ABA dependent and ABA independent pathway are mediators of the signal during osmotic stress. Secondly, temperature stress. Temperatures plays an important role in determining the growth pattern from seed from germination to ripening. The effect of temperature stress on grain development depends on the phase of the crop. At the cellular level, high temperature phenomena can lead to cell system degradation such as malfunction, denaturation, and loss of function. Stress signal transduction path. This is the common signal and pathway. Generally, the signal transduction pathway begins with the perception of the signal. After that, second messenger will be produced. This process begins with protein phosphorylation, whereby the target protein will be directly involved in stress-related genes transcription factors. The product of these genes are involved in the production of molecules that regulate plant hormones such as ABA, ethylene, salicylic acids. Actually, this chart show the direct signal and pathway in plant. This is the common pathway, starting with signals and then receiver, which are receptors. In this chart, calcium and second signal molecules, and then it binds with phosphoprotein cascades, transcription factors, stress response, and then all the response. Abscisic acid, a central regulator of abiotic stress tolerance in plant. Studies on ABA have been conducted over the past 30 years where the ABA signal is said to be a very complex process. One of the first things to note in a stress-prone organism is how plants respond to stress. Any changes in the osmotic phase of cells such as temperature stress, salinity, or lack of water can cause changes in the plasma membrane. Over the past few years, scientists have achieved a great deal of success where ABA receptors in plants have been identified. This discovery has directly solved this complex network. Of all the ABA receptors found, the PYR-PYL-RCAR receptor is the most acceptable. Of all the ABA receptors found, the PYR-PYL-RCAR receptor is the most acceptable. These receptors were isolated in 4 groups using several methods. Structure analysis showed that the ABA binding site was in the hydrophobic space of the PYR1, PYL1, and PYL2 receptor. While this combination occurs, PP2C protein interaction also occur. And in the process begins with the incorporation of start protein and forms the ternary complex consisting of PYR, PYL, RCER. This complex then releases a negative input whereby a signal pathway has been provided by PP2C that disrupts protein kinase SNF1, one of the key components of the ABA signaling pathway. Before this, there have been several studies show that the PYR and PYL protein and the entire PYL family except PYL13 have the ability to activate the ABA signals. This is an evidence and confirms that PYL is an ABA receptor. Further research by other scholars suggests that some PYL are more likely to bind different ABAs. In the early stage of ABA to respond, the synthesis of ABA begins in apical root cell as well as in leaf mesophilic cells. ABA in the root tissue will use apoplastic and symplastic pathway to the xylem. After that, xylem ABA will act as an early signal on to initiate stomata regulation. When water is depleted, ABA biosynthesis in leaves is stimulated by leaf trigger stress. At this time, ABA concentration increase in all leaf tissue and controller cells. ABA leaf synthesis is carried from the phloem to the root, either into the xylem or into the root tissue. ABA mediated signaling begins with the interaction of ABA with the formation of complex ternary combinations. ABA, its receptor PYR, PYLR, CAR, and PP2C. This is actually a diagram or the flow chart uh, about ABA responses and how ABA transport to acting site in a plant cell. Actually, this is a simplified model for abscisic acid generate. So it starts with the ABA biosynthesis induced by environmental cues in the vascular tissue. The newly synthesized ABA is then transported to the cells of acting site for ABA responses by passive or active transports. And then the ABA importers and exporters can efficiently and directly deliver ABA to the acting site. PYR, PYL, RCAR receptors perceive ABA signal intracellularly and then combine with the negative regulators of ABA signals, PP2C, to form the ternary complex. Uh, and then the negative regulator is inactivate, whereas the downstream target of PP2C, SNRK22s are allowed to be activated by the phosphorylation process. The activation of SNRK will thus initiate an ABA response. In cell without ABA, the SNRKs is inactivated by PP2Cs. As I mentioned before, hormone response and pathway work individually or in combination form. There are possible links between hormone signal activated. We call it as a crosstalk signaling. In this case, this is the ABA crosstalk and other hormonal pathway enrolls under heat and drought stress in flowering period. As we can see here on the left flow chart, it show the direct response and pathway for ABA, starting with the drought stress and then ABA dependent pathway receptors PYL and PYR, and then straight to drought responses. Whereas on the right side, right box is the ABA crosstalk under heat and drought stress, which are involved the other hormone such as jasmonic acid and gibberellic acid. Okay, this is also the simplified model of hormone signaling pathway under different abiotic stress combinations. This is also the crosstalk signaling for hormone, the simplest model that suggests the tailored response to, we can see here A, drought and heat combinations involving the jasmonic acid, ABA and salicylic acid for stomatal closure. The B is drought and UVB combination uh, involve the ABA, and then C is osmotic and cold combination. For the conclusion and future prospect, observation and understanding of perception and signal of abiotic stress response can provide valuable information so that new technologies can be generated to reduce losses in the agricultural industry. Further research to identify the signaling response of abiotic stress should be pursued so that new plans and strategies in the agricultural industry can continue to be developed at the same time to reduce the loss of yield from abiotic stress. That's all. Thank you.